All right, I'm going live on Facebook, everybody. Pushing the button now. Hello, everybody, and welcome to the Lunchtime Discovery Series. My name is Chris. I am your host. I work at the North Carolina Museum of Natural Sciences, and it is good to be with all of you again once more for this great lecture series. Every Wednesday at noon, we meet interesting people who are making interesting discoveries all across the world of science, technology, engineering, mathematics, education, nature conservation, all those great topics. I hope that you've all been able to take in plenty of these presentations now that they're here right here on our Facebook and YouTube pages, easy to get to and easy to participate in. Of course, if you can't stay with us the whole time, you can always click the link, come back and watch the whole thing all over again. Also, I'm pretty sure it's going to be a great show. We have a great guest with a really interesting topic. So I think you're going to want to come back and watch it twice just to make sure that you learned it extra. As we go through today's program, remember to drop your questions, comments, experiences even into the chat as we go. That way, when we get towards the end of the presentation and we're ready to pose your questions to today's guest speaker, they're ready for me, queued up. I can grab them out of the chat and pose them to today's guest. Today's presentation is also brought to you by the North Carolina Office of Environmental Education. We all work together to put on this presentation every week and bring you a great show. Many thanks to the folks over at the Office of Environmental Education. Make sure you're following them on Twitter. They're at North Carolina EE. They post great stuff from all across the state of North Carolina. And make sure you check out their website, eenorthcarolina.org, to see more virtual events that were coming up, including this lecture for the rest of June. Now, I'll go ahead and let everybody know. Next Wednesday, when you join in and tune in, don't worry if you don't see me. Next week, I'm going to be out. Carrie Woke is going to be your guest host for the Lunchtime Discovery Series. So if you don't see and hear my voice, don't worry. You made it to the right place. You'll be there. Carrie's going to do a fantastic job. You've seen Carrie before. She does great. Now, let me introduce today's guest to talk to us about some of the things happening in North Carolina's coastal wetlands. Our guest today from North Carolina State University is Dr. Melinda Martinez. Dr. Martinez, welcome to the program.
Yeah, thanks for having me.
I'm really excited to learn more about today's topic. In years past, we've heard from, I think, some of your collaborators at NC State on this topic of ghost forests. So I'm really interested to get your experiences and perspective on this interesting situation in our coasts.
Yeah, I'm excited to share with everybody some of the chapters of my dissertation. Should I go
Fantastic.
To share?
I will let you take it away.
Can you see that okay?
Looks perfect.
Great. Well, yeah, once again, thank you for having me. And I'll just be discussing some of the chapters of my dissertation very shortly, which detail the consequences and early warning signals of ghost forests here in North Carolina. And so this is just like a sketch I drew of me in the ghost forest.
Which is a beautiful illustration, by the way.
Thank you. Wait. Okay, so first off, what are ghost forests? Ghost forests are forested wetlands that were healthy in the past, but are now rapidly transitioning to marshes. And so the term ghost forest is used because The standing dead trees, which are also sometimes called snags, are remnants of its previous state, which was a forest. And so the transition is caused by repeated disturbances, either due to saltwater intrusion, and this could be drought induced, or it could be due to storm surges from hurricane events, but also a different disturbance is increasing flooding events. And sometimes it's kind of hard to tease out which of the two is the main one. And so as the climate changes and sea level rises, we do expect to see expansion of ghost forests along the coast, and not just here in North Carolina, but across the entire southeastern US coastline. And so it's important to understand the changes from forest to marsh, because it means there will be a change in ecosystem services. And so as freshwater forested wetlands transition to marshes, the diversity in vegetation is reduced to more salt tolerant species. And so there's also a change in biodiversity because there's more openness due to the loss of canopy, as you can see in this image. I'll just get my laser pointer. And so understanding the consequences of ghost forests and anticipating transitions is helpful for restoration efforts by focusing on areas that could potentially be restored. And so lastly, the results from this dissertation also help, or from this research in general, help with land management efforts by understanding how carbon sequestration efforts may change from one ecosystem type to another. And so the first portion of this talk, shown in blue, focuses on assessing the consequences of ghost forests in relation to greenhouse gas emissions. And then the second part, shown in green, focuses on anticipating transitions using remote sensing or satellite imagery. So I'll begin with the first section, focusing on greenhouse gas emissions from the standing dead trees themselves, or snags, which is shorter for that. And so the greenhouse gases that I'm focusing on are carbon dioxide, methane, and nitrous oxide. And these are important because of their ability to absorb energy. So these three gases are found in the atmosphere, but at different concentration levels. So carbon dioxide, or CO2, is more commonly known in the atmosphere. It's about 400 parts per million, or ppm. And so while methane is about 2 parts per million, and nitrous oxide is about 0.3 parts per million. And so although methane and nitrous oxide are much lower in concentration in the atmosphere, they are more potent than CO2. So methane is about 40 times greater than CO2, or more potent than CO2, and nitrous oxide is about 300 times more potent than CO2. And what this means is that methane and nitrous oxide have the ability to absorb more of the sun's energy, which keeps the earth warmer, acting as a greenhouse would, which is why we use the term greenhouse gases. And so globally, wetlands occupy a very small portion of the landscape, but they do contribute significantly to the carbon cycle through carbon sequestration. And so this means that they store a lot of carbon in the vegetation biomass, like in the trees, in the plants, and then in the soils, but they also, the vegetation uptake of CO2 through the vegetation itself. But wetlands also naturally produce a lot of methane. And so wetlands are known as the largest natural producers of methane, especially freshwater wetlands, contributing as much as 20 to 30% of the global methane emissions, which is a lot. And then more recently, studies have focused on tree stems as an additional source of greenhouse gases that could provide more insight into the global carbon cycle, but also the nitrous oxide global budget. And so in forested wetlands, there are various paths greenhouse gases can take to be emitted from the soils to the atmosphere. And so methagenesis, which is the production of methane, occurs in soils because in wetland soils because of low oxygen conditions from high water levels and constant soil saturation. So nitrous oxide can also be produced in the soil. And this is due to incomplete nitrification and denitrification. So it's like a byproduct. So these soil greenhouse gases can be emitted directly to the atmosphere, either by diffusion or through ebullition. And so diffusion means it travels slowly through the soil, through the water column, and is emitted out to the atmosphere. But because it happens so slowly, especially for methane, there is a potential that methane can be oxidized, which means it's consumed and then by microbial communities and then converted to CO2 and then is emitted out to the atmosphere as CO2. And ebullition occurs, this is just like bubbles. This occurs when gases accumulate in the soils and they just bubble to the surface. And so the greenhouse gas concentrations inside these bubbles are much higher because there is a chance for oxidation because it's not as slow as diffusion. But the last transport mechanism is through tree stems. And in live trees, this occurs because of the movement of soil water up the tree stem. And so the gases are able to be emitted through this same transport mechanism and through the sides. But in standing dead trees, a lot of the water inside the trees is flushed out, leaving a network of open cells, which allow greenhouse gases in the soils to move to this concentration gradient. And so in a way, these standing dead trees are acting as straws for greenhouse gases produced in the soils and are emitted to the atmosphere through the sides. And so here in North Carolina, I've shown in this reference map, there are many freshwater forested wetlands that are transitioning to marshes along the Albemarle Pamlico Peninsula, which I refer to as the APP. And so I selected 5 sites around the peninsula that are experiencing various levels of salinization and flooding. And so wetlands on the northern portion along the Albemarle Sound, such as palmetto pear tree, are more fresh, so salinity levels are about 0 to 1 parts per thousand, as opposed to wetlands on the southern portion of the peninsula along the Pamlico Sound, whose salinity levels can be as high as 15 parts per thousand. And so this includes sites such as Swan Quarter and Gull Rock. And then Pocosin, this site in the middle of the peninsula, is right along the Intracoastal Waterway, and this is considered a fresh site. And then Point Peter is considered a brackish site, which means it's about up to 6 parts per thousand. This is in between the Albemarle Sound and the Pamlico Sound, but right off the Croatan Sound. So it is considered more brackish. But within this peninsula, there have also been many canals and ditches created to drain the land to be more conducive for agriculture. As you can see, these lines, these are canals that are currently on the peninsula. But because there's so many, there's a lot of hydrologic connectivity. And so which means some of the brackish water from the Pamlico Sound, it can travel further inland into these freshwater ecosystems. So what we're seeing is forest dieback further inland in areas where you wouldn't expect. And so the main questions I wanted to ask were, do snag stem greenhouse gas fluxes differ from nearby soils? And so if you're not familiar with the term flux, this just means the movement of gases flowing in and out of a very specific area. And just as a reminder, snags are standing dead trees. I just use this interchangeably. And so the second question, are there differences in snag stem greenhouse gas fluxes across the peninsula? Because there are differences in salinity and flooding from north to south. And so this is the general monitoring setup I had for each site. The field campaigns occurred during the growing season from May to September in 2018 and 2019. And then so for each site, I selected 5 snags, and snag stem greenhouse gases were measured using this flexible chamber that was attached to the tree and sealed with mini zip ties. And then about 1 to 2 meters away, soil chambers were also installed. And so greenhouse gases were measured in 2 ways, using a gas met, which is a portable gas analyzer, or the syringe method, which means you just take a sample from within the sealed chamber through a rubber septum and then bring back those samples to the lab. And so fluxes were calculated by the greenhouse gas concentrations building up. So as the greenhouse gases build up in these closed sealed chambers, we measure the concentrations over time and divide that by the time of seal, and that's how you calculate a flux. But we also took weather parameters, and then in 2019 for each site, I installed a water monitoring well for each site, so 5 wells in total, and each well had a water level logger and a conductivity logger, and this just lets us know what the salinity levels are within the soil water table. And then in 2019, I also installed pore water sippers, at two depths, 15 and 30 centimeters. And this is just made out of PVC with tubing attached to a pumice stone at the bottom to extract some of the pore water, the soil pore water. And so this is just some of the images showing what the chambers look like in real life. This is me collecting greenhouse gases from the snag stems, and this is the soil chamber. And this is a portable gas analyzer. And so for the next few slides, I'll be showing each respective greenhouse gas flux. This slide showing CO2 fluxes for snags on the left and then soils on the right. And this is by year and site. So freshwater sites, which I'm considering 0 to 1 parts per thousand salinity level, are in these warm colors. The brackish site, Point Peter, which is 3 to 6 parts per thousand in gray. And then more saline sites, which I'm considering 6 to 15 parts per thousand, are in these cooler colors. And so one thing that's very important to note is that in 2018, which is indicated by this vertical dark blue bar, 2018 was a much wetter year. And so during the growing season, cumulative precipitation was about 896 millimeters, while in 2019, which is considered a more drier year, for this same time period, it was about 666 millimeters. But the precipitation levels for 2019 were closer to the overall median for this area, which is indicated by this dashed gray horizontal line. And so going back to this, so CO2 fluxes are on the y-axis and the years are on the x. And overall, we did see that snag stem greenhouse gas fluxes were 4 times less than the soils. And you can see this by simply looking at the y-axis of these between the soils and the snags. And so for soils, it could be as high as about maybe 800 milligrams of CO2 per meter square per hour. But then for snags, it only goes as high as like maybe 250. And so for snags, we didn't see very many differences between years, even though there were differences in precipitation levels or in sites. But for soils, we did see significant differences for among sites and across years. And we saw the higher fluxes in 2019, saw higher CO2 fluxes in 2019, which makes sense because these are the drier conditions. So there's more oxygen present in the soil. And so this slide shows the methane fluxes. So once again, snags are on the left, soils are on the right. But for snag methane fluxes, we didn't see any differences between years or among sites, and there was lots of variability both years. But for soil fluxes, however, we did see significant differences by year and a slight interaction effect. But there was higher variability in 2018 in the soils. And this is during the wet year, which is what is expected because there's 0 oxygen conditions in the soils, which is conducive to methane production. And then 2019, this was greatly reduced because it was much drier, and so there's more oxygen present in the soils. And so lastly, these are the nitrous oxide fluxes. And for the snags, we did see a significant difference by year and site, but there was a lot of variability both years. And then in soils, we did see significant differences by sites and a slight interaction effect. And this is likely because for Point Peter in 2018, this was more of a nitrous oxide sink, which means nitrous oxide was being consumed. And in 2019, it was still considered a sink, but it was greatly reduced. And so when we go back to our questions, do snag stem greenhouse gas fluxes differ from nearby soils? We can answer yes. Soil greenhouse gas fluxes were 4 times greater than snag greenhouse gas fluxes, but when we go back, yeah, but snag stem greenhouse gas fluxes are often not considered at regional or global estimates. So I guess a different way of viewing these results is that snags contribute an additional 25% of greenhouse gases unaccounted for from standing dead trees alone. And so for our second question, are there differences in snag stem greenhouse gas fluxes across the peninsula? Because they do vary in salinity and water levels. We can answer no and yes only for snag nitrous oxide fluxes. And so what this further tells us that snag greenhouse gas emissions are not as predictable as soil greenhouse gases, which are strongly driven by water levels. And this could be because the snag root structure is still intact, possibly transporting gases from deeper soils than what we were measuring. And so moving on to the second portion, this focuses on identifying the main sources of methane, specifically in the standing dead trees or snags. So previously I said that these greenhouse gases, but more specifically methane, is coming from the soils through this pathway from the soils and being emitted. But because these trees are dead, it's also possible that methane can be produced internally just as the tree is decaying over time. And so the monitoring setup is the same as the previous section, except I also sampled gases from within the tree by drilling inside at 3 different heights, 30, 60, and 120 centimeters, just to see how the concentrations of methane change with increasing stem height. And then I also extracted additional gas samples from the snag and soil chambers, and then also extra samples from the 60 centimeter height for carbon and hydrogen stable isotope analysis. And then I also extracted extra pore water samples to determine how much methane was diffused in the soil pore water, but also for stable isotope analysis. And so if you're not familiar with stable isotopes, these are elements that are stable, unlike radioactive isotopes that decay over time. And so most biological elements like carbon and hydrogen have 2 or more stable isotopes, which mean that they have the same number of protons, but different number of neutrons. And so the more neutrons you have, the heavier they are. And so they become less energy efficient. And so microbial communities actually discriminate against carbon-13 and they prefer this lighter isotope. And so what we're measuring is the ratio between these two, between the light and the heavy isotope. And so if microbial communities consume more of the lighter isotope, there'll be more of the heavy isotope left, making the ratio value higher. And so stable isotopes are useful in this way for tracing, sourcing, and recording changes in these elements throughout different ecosystem processes because the ratios change during these different processes. And this includes oxidation, which means the consumption of methane and it's converted to CO2. And so here I'm showing the methane concentrations from those 3 heights, 30, 60, and 120 on the x-axis. And then note that the methane concentrations is log scaled. And just as a reminder, methane in the air is close to 2 ppm. But in general, what we see is a decline in methane concentrations across each site with increasing stem height. And so this graph shows the carbon and hydrogen stable isotopes from the different sources measured. Hydrogen is on the x-axis, the hydrogen isotope, increasing in weight from left to right. And then the carbon stable isotope is on the y-axis, increasing in weight from the bottom to top. And so the circle data points here are individual observations, and the squares are average per source type, which are the pore water, the soil chamber, snag chamber, and inside the tree themselves. And then lastly, air samples. And then these lines are just the point distribution of the individual observations. But what this data shows is that the pore water samples, which is in blue here, have the lightest carbon and hydrogen stable isotopes. And this increases in heaviness when it goes to the Heartwood, the soil chamber, snag chamber, and then it's heaviest in the air. And what this means is that methane from these sources is getting more oxidated, which makes the isotope heavier. And it means that this is likely the path going from the soil pore water through the Heartwood out through the snag stem, which is what we measure in the snag chamber. So in summary, we saw that methane concentrations decreased with increasing stem height, but also the carbon and hydrogen stable isotopes were lightest in the pore water and increased in heaviness when it goes through this pathway. It is heaviest through the snag stem. But then it increases in heaviness because it's being oxidized throughout this process. And so what this means is that the standing dead trees, which we previously thought as straws for soil produced gases, are really more like filtered straws, because the methane produced in the soils travels through the snags, remains really high in concentration in the heartwood, but it's being oxidized as it makes its way through the snag stem before it's being emitted out to the atmosphere. And so the porosity in the snags, which is determined by how decayed the snag or standing dead tree is, might be what determines that amount of oxidation or consumption of methane. And so moving on to the last section, this focuses on detecting early warning signals of coast to wetland transitions, including ghost forests. So some of the objectives in this section were to first determine the capability of detecting early warning signals, using remote sensing, imagery, and analyzing vegetation changes over time. And then I applied this time series analysis across the peninsula to determine which areas might be approaching a critical transition. So critical transitions are defined as changes in states from, they're defined as changes in states. So here the first state is a freshwater forested wetland, a healthy freshwater forested wetland, and the second state is a marsh. And so tipping points can be kind of difficult to determine because an ecosystem can show very little change before the tipping point is reached, but one way to determine the potential of a critical transition is by assessing the loss of resilience. And so this ball and cup potential model demonstrates how environments change from one state to another based on loss of resilience. And so by resilience, I just mean the ability of a system to recover after disturbance. And so this system on the left has very high resilience, which means that if this ecosystem experiences a disturbance, it pushes that ball closer to that tipping point, but because it's so high, it can recover fairly quickly, and this ball can go back to its previous state closer to the base of this cup. But over time, if this system experiences many disturbances, repeated disturbances, or extreme disturbances, the resilience can be reduced, and so that even a small disturbance can push this system into the new state, which will be the marsh. And so I determined loss of resilience over time by focusing on areas that have already transitioned and going back in time and seeing if I can detect a signal before the critical transition. And so one of the ways we can study changes in environmental states is by using remote sensing. And so I used the Landsat archive, which starts from 1985, and it continues today, but I went up to 2020. And this is at a 30-meter resolution. And so in Google Earth Engine, I created yearly max NDVI composites during each growing season. And so if you're not familiar with NDVI, this just stands for Normalized Difference Vegetation Index, and it's commonly used to assess the health of vegetation. And so this is just calculated by the difference between two wavelength bands that are captured by the satellites. And so this figure shows what I expect to see in the time series in relation to resilience with NDVI. And it's important to note that forested areas have a higher NDVI just because they tend to be more green than as opposed to marshes, which have a lower NDVI, which is indicated by this horizontal brown bar. And this is just because they tend to be more brownish. And so for highly resilient areas, I expected there to be very quick recovery after disturbance. And so disturbances here are noted by decreases in NDVI, but also these vertical gray bars. But you can see that it gets back to its previous NDVI value after the disturbance. But in low resilience systems, you expect that there would be a longer recovery period after this disturbance. And this is a period that we refer to as critically slowing down, which is an early warning signal that at the next time this system experiences a disturbance, it might get pushed past that tipping point, which is indicated by this vertical red dashed line into the new state, which is the marsh. And so I used I selected very specific areas of known transitions, but I also applied the time series analysis across all natural wetland ecosystems along this peninsula. I did this using a high performance computer. And so for each time series pixel, I analyzed the standard deviation change over time using a 6-year moving window. And so these are the same NDVI graphs from before for high and low resilient. And then so I just wanted to see how the variance or standard deviation changed by comparing these 6 years to the previous 6 years and seeing how that changes basically over time. And so for highly resilient areas, I expected the standard deviation to remain fairly low. As opposed to the low resilient areas, I expected the standard deviation to increase over time. And this is because it's taking longer to recover, which means it's not looking the same as previously before. And so there's just a lot of variance happening in low resilient as opposed to high resilient systems that recover fairly quickly. And so I also looked at the overall correlation trend of the NDVI to get a better idea of which direction the ecosystem was headed, whether it was increasing, remaining the same, or decreasing, but also getting the correlation trend of the standard deviations. And so I started off by assessing very specific areas of known transitions or lack of transitions. And so I looked at images from 1985 and selected areas that were forested in 1985 and are still forested in 2020. And these are forest to forest in this dark green. And then I selected areas that were forested in 1985 and are still forested today, but are now adjacent to marshes. And so these are areas that would be next in line in transition. And so this is forest to edge. It's in light blue. And then I selected forested areas in 1985 that have transitioned or are currently transitioning. So this could be a ghost forest. And these are in these yellow points, forest to marsh right here. And then lastly, I looked at marshes or areas that were marshes in 1985 that are now open water. And so this occurs more along the shore. These are marsh to water red points here. These are more along the shoreline, and this is likely due to shore or erosion. So these are the average time series from those selected transition types. And just note that NDVI is times 1000 here, but this is from 1985 to 2020. And what we see is that for each transition, it kind of played out as expected. And so for forested areas, the NDVI is fairly high. And then you can see in 1993, there is a disturbance event, but it does recover fairly quickly and remains fairly high throughout the time series. And for forested areas that are now on the edge, adjacent to marshes, we see that same decline in 1993, quick recovery, but then starts declining over time. And then from 2010 to 2014, we do see this massive decline. And this lets us know that these areas are likely, this is the critical slowing down period, and it's very likely that they will be transitioning next. And so for forest to marsh, we do see that the NDVI, which is this yellow line, the NDVI is much lower, slightly lower than the forest to forest, but the same, disturbance event in 1993, but it just continues declining after that. And this lets us know that the resilience from the start of the time series for some of these areas was already likely very low. And so the same pattern occurs for the marsh to water transitions, although the NDVI is much lower, and it's because marshes have much lower NDVIs in general. Okay, so here I'm showing examples of the early warning signals for each transition type. The top panel is the time series with the Gaussian filter, which is this red line. In the middle panel is the residuals or distance from these observations to this red line. And this just gives us a better indication of variance and how that changes over time. And then the bottom is the standard deviation with that 6-year moving window. And so for the forested areas, we can see that the standard deviation declines over time. The NDVI remains fairly high as well. And for areas that were forested or now on the adjacent, we can see that it's taking a little bit longer to recover after disturbance events. We do see an increase in standard deviation just for these areas, which are still forested today. Oh, you can see that. Sorry, forgot about that. And then so for these areas that are now in the process of transitioning, we can see that we see that same increase in standard deviation just prior to that critical transition, which is right around point 25, which is 2010. But we do see the increase in standard deviation prior to that critical transition. And this occurs also in the marsh to open water, where you see that critical transition occur right around this time point. We do see an increase in standard deviation. And so we can see here that the standard deviation can be used as an early warning signal for critical transitions, and it can at times even be detected up to 10 years before this critical transition point. But there also is a chance for false positives and false negatives. And so here I'm showing the same transition types with similar NDVI trends at the top, but opposite standard deviation trends at the bottom. And so, for example, the forest to forest transition here on the far left, the NDVI remains fairly high, but now the standard deviation is increasing as opposed to the previous slide that was decreasing. And this is an example of what is considered a false positive, which means that an early warning signal is picked up when there shouldn't be. So this is a false positive. And so here in this next one, for these next three transition types, we do see the opposite of the previous, where previously we saw an increase in standard deviation, but now we're seeing a decrease in standard deviation. And these are examples of false negatives where no signal is detected, even though there are critical decisions happening. And so this happens across all three. And so in order to help avoid some of these false positives and negatives, I combined the standard deviation trends with the overall NDVI trends to generate these four possible scenarios. The top left, which shows a decline in NDVI and an increase in standard deviation, means an early warning signal can be detected and intervention can take place. And second, where we have a decline in NDVI and a decline in standard deviation, and this means that no early warning signal can be detected because the transition is happening so slowly. And then the third is where you have an increase in NDVI and then an increase in standard deviation at the same time. And this occurs for areas that are maybe recovering or being revegetated, so becoming more green over time. And this is following a disturbance event, which is why we have such high standard deviation. And the last scenario is where you have an increase in NDVI and a decrease in standard deviation. And this is for areas that are becoming more stable over time. And so no management is needed for these 3 and 4, but management can take place for 1 and 2. And so across the peninsula, I combined the NDVI and standard deviation trends showing the 4 scenarios I previously mentioned. And so the dark green here is the increase in NDVI and decrease in standard deviation. And so these are areas that are stable ecosystems. And the light green is areas that are increasing NDVI and increasing in standard deviation. So these are areas that are being revegetated, recovering, becoming more green over time, following a disturbance. And then the red are areas where we can detect an early warning signal because the NDVI is decreasing, but the standard deviation is increasing at the same time. And then the yellow is areas that are where the NDVI is decreasing, so it's becoming more marsh-like, but the standard deviation is also decreasing. So these are areas that are just gradually transitioning. And so the boxes on the right are just zoomed in areas from the sites from the previous 2 sections. But the top left, this red box here, this is showing a restored wetland. And what we can see is that most of the restored wetland is now considered a stable ecosystem, although some of the areas are on the decline. These are areas that are experiencing more flooding within that restored wetland. In this middle panel, as you can see here, this is at Pocosin site right along the Intercoastal Waterway. And what we see is that there's a lot of red here, which is the early warning signal. And I've personally driven by throughout this peninsula and seen a vast amount of snags. So like a ghost forest definitely is present here. What else? Oh, and then this middle, so here in this section right near Phelps Lake, you can see this light green. This is an area that's being revegetated. And this makes sense because this area experienced a fire in about 2008. And so it has been recovering from that fire disturbance. And so these results kind of make sense, are more very in line with what we know that is happening on this peninsula. And so in conclusion for this area, for this section, we know that we can detect early warning signals up to 10 years prior to a transition, but we need to watch out for false positives and negatives. And across the landscape, was able to determine that 40% of the wetlands are actually stable, while 14% are showing signals of an oncoming critical transition. And then just to sum up, going back to the first section where I determined snag greenhouse gas fluxes are not as predictable as soil greenhouse gas fluxes, and that snags could contribute an additional 25% to the ecosystem. And then for the second chapter, we determined that snags are actually acting as filtered straws and converting some of that soil produced methane to CO2. And then what I just showed here in this last section is that we can detect early warning signals of ghost forest up to 10 years prior to a transition. But of course, there's false positives and false negatives, and that up to 14% of the peninsula is or will experience a critical transition in the next few years. And so, of course, I'd like to thank my funding sources for this. And if you have any questions, feel free to email me, or you can follow my fieldwork adventures on my Twitter.
Dr. Martinez, thank you very much. Excellent presentation. Everybody, big round of applause wherever you're at for our guest. Very interesting stuff. I'll be honest, having heard just a little bit about this in the past, not much, but just learned a little bit about it. I expected to see on your maps, for example, huge swaths of coastal forest turning into snags and marshes and ghost forests, as the terms go. So it was interesting to me to see that there are parts of our coast that are reforesting and revegetating in a healthier state.
Yeah, so this is like this is what makes ghost forests like these transient ecosystems. So they're not always going to be a ghost forest. It's going to eventually become a marsh. And sometimes this happens gradually or more quickly. And I think that's kind of what's unique or very cool about what this last section shows, is that these processes happen both slow and fast within the same peninsula.
What was it like conducting this research out in these coastal environments? Like something about, like I love a wetland, but something about like walking through them for probably several hours a day with lots of electronic equipment just seems, one, very adventurous and exciting, and also it had to have been hot.
It was so hot. There's so many mosquitoes, so many deer flies. It's definitely, I definitely think working in wetlands is among the hardest field work because it's constantly hot, very humid, and there's so many mosquitoes, so you have to like double layer while you're already like sweating. Sometimes you have to wear waders, sometimes chest waders, because you don't know how high the water is going to be, and so there's like not much ventilation in the chest wader. So yeah, it's very hard work, but someone's got to do it.
Somebody's got to do it. Well, I mean, just the research is very interesting. The way that a dead tree is just siphoning greenhouse gases up into the atmosphere and contributing to climate change in that way, and that the problem itself is stemming from, I think I understand, from human causes already. There's the irrigation canals,
Yeah.
and then there's climate driven things like sea level rise.
Yeah, it's definitely a positive feedback loop. So it's just, and that's why we're expecting these areas to keep moving. Like we're expecting the marshes to migrate, and as the marshes migrate, they're going to take over some of these forested areas, and that's where you see those ghost forests. And that's why there's such transient areas. So that's why you don't see. That's why the whole peninsula isn't like you didn't see the whole thing like all in red. It was just very like small slivers along the shoreline.
Gotcha. Excellent. Well, let me see what people are saying over here in the chat and throw some questions your way. All right, the first one I've got here. Are there studies on the impact of dumping in the ocean and sea level rise, sinking ships, whether on purpose or not, concrete material dumping for reef builds, or just trash in general? Do you think any of those human impacts would be involved in what you study?
I think it'd be kind of a stretch, and it's definitely outside my expertise. So, and I know that there are like a lot of rig to reef programs, especially in Texas, along the Texas-Louisiana coast, but that's the extent to my knowledge.
Okay. We'll have to get another expert in here
Yeah.
on that one in the future. Okay, next one from, let's see here. If ghost forests is specifically the term for former forests that are in areas that are now marshlands, is there also a specific term for former forests that drowned in freshwater settings?
Yeah. So, I mean, I guess if you can still see the snags standing in the trees, that would definitely still be considered a ghost forest. And I think, I use the term ghost forest here, but when there's like outbreak, like beetle outbreaks, maybe out in the Midwest, you can see like vast majority of snags. That would also be considered a ghost forest. It's just like where you see a lot of forested areas that have just quickly died. And it's just so many that when you see it, you're like, something must have definitely happened here for this many trees to have died off. But we do see that on the coast here where there's you can see snags standing in the trees right along the shoreline. So they're like in water, but the snags are still upright, which is kind of cool to see. But that would still be considered a ghost forest.
The name seems fitting too, like the animation you had at the beginning of the show, the drone footage flying over, and it's just dark and light gray stands of trees, and like the landscape looks ghostly,
It's like very
abandoned.
eerie.
Yeah.
And that was actually footage from the Science Friday that we the podcast that we did about in 2018.
I'll try to find a link to that and drop it in the video description after the program. So everybody come and check back and look at that because it's a really great video. Okay, let's see. Holly's got a question. Given that so many housing developments have overtaken some of our wetlands, would this actually help our wetlands, even though it's bad for the forest?
Now, and many times when there's human structures in place along the shore, it doesn't help the marshes migrate. So the marshes migrating, it just happens naturally and happened long before even like, especially during the Holocene era, like when the water sea level rise was increasing and decreasing. And so with that, you get some of that marsh migrating inland or prograding, like maybe expanding. But when you have structures in place, it kind of like stops the migration at that point, and so it doesn't help. Like we need the marsh needs the space to migrate.
And once you put a house there, we say no more marsh and no more forest.
Yeah, you're just removing all ecosystem services.
And then a follow-up. Holly was trying to find any way to view this as balancing out because Holly's looking for some good news. Is there any silver lining?
Okay, so I guess ghost forests they don't always have to be viewed as a bad thing because they do become, I know I talked about this shift in ecosystem services, but and I call it a shift and not a decline because sometimes they do become new habitat spaces for other species that like more open canopy. And so, I think some people talk about like, well, what should we do for these ghost forests? Like, should we cut the trees down or what? And it's hard to answer that because they do become new breeding grounds for some areas, for some species, for bird species. And so I don't know, it's difficult.
Other stuff moves in and takes over.
Yes.
Okay. Well, there you go. There's habitat. I guess it's kind of like climate change. It's the rate of these transitions maybe that are the reason, for example, that you're trying to find an early warning signal.
No, absolutely.
All right, let's see here. Here we go. What is the mechanism whereby snags convert methane to carbon dioxide?
That's just microbial communities consuming the methane, so they're using it as an energy source. And during that digestion process, I guess it gets converted to CO2.
There we go. Thanks, Glenn. And Andrea watching on Facebook asks, do you work with a team of students and or researchers in gathering the data? And if so, how many were on your team?
Yes, I definitely worked with many undergraduates. I think it was a total of 4 or 5, four undergraduates during the span of 3 years. And then of course my advisor. But yeah, like usually like for field work, you only need like one other person. You don't really need more than 2. Becomes like too many people because it's a lot of waiting around because you're waiting for these gases to accumulate in these chambers. So it's just waiting in the hot, wearing chest waders.
Standing out in the sun, just
Yeah,
waiting on those trees to fart.
yeah, basically.
I saw a tweet from the Environmental Education Office where today we were going to be talking about gas exchange and tree farts, and I thought that's an interesting way to put it.
I mean, it is because we like when you think about your body digesting, you have microbial communities inside your stomach that are digesting your food, and then the gases that like the gas is a byproduct, and so that's what you kind of release. It's a slow release within your own body, but it's also happening a slow release in the trees from the sides because of microbial communities.
Were you ever standing in a wetland and just see some bubbles come up and think, oh, the tree just exchanged some gas?
I think when you're walking, you can definitely see the bubbles, but that's like when you're forcing those bubbles to come out. But yeah.
Sure. Okay. This looks like it might be the last question that's here so far. Folks, we got a few more minutes, so if you got more questions, drop them in the chat. What are the maintenance techniques that can be done on those red areas?
So, yeah, I'm not entirely sure what exactly can or should be done. And I think for this, at least the research that I've done so far, I just wanted to, provide stakeholders with at least a guidance of like, hey, these are the areas that maybe restoration efforts should be focused on. And this could be maybe facilitating more the marsh migration or maybe planting new trees that are more tolerant to salt water or increasing flooding. But I think I would have to do more research or read more to figure out exactly what needs to be done.
It seems like a very complicated issue to try to manage. Unlike managing like a more terrestrial system with saltwater intrusion and flooding and sea level rise, that's so many more factors, at least in my mind, than, is this pine forest outside of Raleigh growing or not growing?
No, it definitely is. And there is water control structures set in place around the peninsula to try to, I guess, reduce the amount of salt water flowing further inland. But but I think like during storm events, there's only so much that water structure can do.
Yeah, sure thing. And I know there are wildlife managers probably watching this who got very upset that I just dissed pine forest management. It's not my intention at all. But this, I know this stuff is pretty complicated, especially since so much of these areas may or may not be publicly owned. I don't know.
Well, a large portion of the
what
peninsula
ownership
is
like.
either state or federally owned.
Okay.
But there's a lot of agriculture also in the peninsula, which kind of makes it more difficult.
Understood. Let's see. Karen's got a question. Were you able to find similar research being done in other parts of the world, like Bangladesh or parts of Southeast Asia or other areas with large river deltas?
I think as far as ghost forest stuff, I mean, this is happening all over the southeastern US, but other parts of the world, I'm sure there is. I may not be as familiar. But with tree stem greenhouse gases, that's becoming more like more and more researchers are realizing the importance of looking at tree stem greenhouse gases. And a lot of people are looking at live tree stems, but I think this is one of the few studies that is looking at standing dead trees, because so much focus has been on live trees. But now that we have these vast amount of standing dead trees, it's still important to look at these too.
Next one, would the forecast of 10 years be appropriate to consider as a possible reforestation?
Yeah, maybe. And like, I think I was hoping that would be sufficient enough time to get targeted restoration efforts. But who knows?
Yeah, you have to, that's the next experiment. Somebody's
Yeah,
got to
we
get
have
out
to
there
actually
and start
do a
planting some
restoration
trees.
effort. Yeah.
Yeah, wow. So interesting. Okay, it looks like we're about out of time, and it looks like I might be out of questions as well, which is right on time. So, Dr. Martinez, thank you. This was fabulous.
Yeah, thanks for having me.
Excellent job. Everybody, thanks for tuning in to today's Lunchtime Discovery Series presentation. We, of course, will be back here next Wednesday at noon. Make sure that you're following the Office of Environmental Education on Facebook and on Twitter. They're at North Carolina EE on Twitter. Check out their website, eenorthcarolina.org. There you can see the schedule of events like the program that's coming up next Wednesday at noon. I'm sure you won't want to miss it. I will be missing it, and I will be thinking of you all next Wednesday at noon. But then I'll be back with you ready for another presentation. So I'll miss you all next week. Think of me while I'm gone. Make sure you're following the Museum of Natural Sciences on Facebook, Twitter, and Instagram. We're at Natural Sciences on all three platforms. And here on YouTube, of course, you can subscribe to the museum's YouTube channel. Click the bell to get notified when we're bringing you great new live video just like this one. Listen, we do several fun, interesting, and exciting live programs every single week. For example, tomorrow night at 7 o'clock, we're actually going to be talking to museum paleontologist Dr. Lindsay Zano, who will be live from a dinosaur quarry in New Mexico. That's right. We're going to be having a live Zoom call from the place where they are right now digging up duck-billed and predatory dinosaurs in New Mexico. So don't miss tomorrow night's episode of Science Tonight. Everybody, take care, stay safe, continue to keep your communities safe. We'll see you again real soon. Bye, everybody.